Hello everyone. So my name is Hippolyte Guerineau and I'm working at Versailles Hospital. And I'm going to present our research experience using optical genome mapping on multiple myeloma samples collected at the Versailles Hospital. So OGM has been implemented in the lab for over a year for myeloma samples. So first, I'd like to introduce you to our workflow. So we collect two bone marrow samples of approximately 4 ml, one in EDTA and one in heparin. If plasma cell infiltration is less than 80%, we perform plasma cell isolation to be able to detect all relevant abnormalities. Otherwise, we do not. Note that we may mix the EDTA samples with the heparinase 1 if necessary. Next, the seg count is performed. So if there are fewer than 0.5 million cells, we do not perform OGM, but instead 3 FISH analyses are run. So FGFR3 IgH, TP53, and 1P, 1Q. If they are between 0.5 and 1 million cells, we dilute the sample with negative fraction to achieve at least 50% of plasma cells, according to the paper from Hélène Guermouche and her colleagues. So indeed, to perform OGM, it's necessary to have at least 1 million cells. However, the study has shown that by diluting the positive fraction containing plasma cells with the negative fraction by up to 50%, it's still possible to detect all relevant findings. If there are more than 1 million cells, we don't dilute with negative fraction. And if there are more than 2 million cells, we also prepare our backup samples. So finally, we extract and label the DNA. We perform OGM for structural variation analysis of the samples, and FISH TP53 is performed to determine the proportion of cells with TP53 loss and to detect minor subclones. So we primarily use guided assembly rather than RVR, as the 1114 translocation was not consistently detected, even when all features were removed. So we evaluate multiple myeloma samples using two previously published research frameworks. So the IFM-based approach, which considers a TP53 loss in more than 55% of plasma cells, and the IMWG 2023 framework, which includes TP53 loss in more than 20% of plasma cells. For analysis, we use the Bell file developed by the Frog myeloma group, and each sample is independently analyzed by two operators, and only relevant findings are reported. Following analysis, ultra-high molecular weight DNA is transferred to the molecular biology labs for TP53 mutation analysis in accordance to the IMWG 2023 framework. Now let's talk about our one-year prospective study. So between October 2024 and October 2025, we received samples of 213 myeloma samples. So most samples were collected at first sampling, over at subsequent time points. Among the 213 samples analyzed, most underwent plasma cell isolation without dilution, meaning there are more than 1 million cells. And in terms of metrics, most of them were optimal. Now let's focus on the different abnormalities. So we observed a slightly higher frequency of IgH rearrangements compared to previous reports, specifically 1114 and translocation. Other IgH rearrangements were observed at frequencies comparable to published data. The frequency of hyperdiploidy, defined as the gain of odd-numbered chromosomes, was similar to published report. Regarding chromosome 1 abnormalities, we observed a slightly lower frequency of 1q gain compared to published data. So detection of 1q gain may vary depending on sample collection time, so baseline versus subsequent samples, and sample size. So a published study often use a 20% cutoff, though criteria vary. A frequency of del1p32 was comparable to published report, and proportion of MYC rearrangements was similar to published data, with most partners being chromosome 14 with IgH, chromosome 6, chromosome 1, and chromosome 22 with IgO. Overall, the distribution of the abnormalities and categories match those reported in IFM and IMWG 2023 criteria. Now let's focus on TP53. So TP53 loss was detected in 18 samples. In 14 of these samples, clone shards were comparable between OGM and fish. Among these, 2 cases with TP53 loss in around 20% of plasma cells were only visible after removing all features. In 4 cases, discrepancy in clone size were observed. So for one, fish detected a larger clone than OGM. However, in this case, TP53 loss affected over 55% of plasma cells, which did not alter the classification of relevant findings. In three cases, OGM detected a larger clone size than fish, including one case that will affect finding under the IMWG 2023 criteria. So in conclusion, TP53 remains performed to assess clone size threshold, as they are not yet been fully validated using OGM alone. Nevertheless, these results are very promising. Now there are other more recent findings apparent thanks to OGM. So a study by Stong and his colleagues investigated the chromosome breakpoint in 414 translocation. So the result indicates that the distribution of breakpoints position relative to the NSD2 gene varies across samples. So breakpoint located before the NSD2 gene associated with one pattern. Breakpoint at the start of the gene fall in a distinct pattern, and breakpoint within or beyond the central portion of the gene define another pattern. So thanks to the bed file created by Frog Myeloma group, we are able to evaluate these breakpoints more precisely. However, it's not always clear-cut, as some breakpoints may fall between 2 defined regions. Moreover, complex structural variants, notably chromoanagenesis, are found in around 35% of samples. So in myeloma samples, chromatosis has been reported as an relevant finding. However, it has been characterized with whole genome sequencing. So therefore, further research are required to better understand its role and significance in myeloma. So there is a group at Frog whose role is to characterize these complex events. So here are a few examples with a circle plot that may suggest a chromatosis of chromosome 13 and maybe 8. Here's one that may suggest chromoplexy. Here, a very complex structural variants with possibly several phenomena at the same time. Finally, OGM can be used to analyze structural variation affecting genes relevant to immunotherapy targets, such as TNFR SF17, which encodes for BCMA, and GPRC5D. So published research reports structural variation at TNFR17 and GPRC5D, including biallelic deletion events observed following targeted immunotherapies. So in public research, monallelic loss of TNFR SF17 has been reported in approximately 9% of multiple myeloma samples prior to immunotherapy. In our dataset, the observed frequency of monallelic loss was lower, around 2%. So here an example of a plasma cell leukemia sample with a very complex optical mapping and with a monallelic loss of TNFR SF17. Moreover, monallelic loss of GPRC5D, a gene of emerging interest in immunotherapy, was also observed in 11% of baseline samples. So in conclusion, in multiple myeloma, OGM is concordant with FISH and provide additional information such as breakpoint analysis, chromoanagenesis, and immunotherapy-related genomic variation. So OGM is successfully implemented in the lab for myeloma samples. So thank you for your attention, and I'm happy to take any question.